Hello, I'm Hannah Donnett with the Collaborative on Health and the Environment. She enjoys bringing you the latest environmental health science through our partnerships, calls, webinars, science serves, publications, and social media. I would like to welcome everyone to the fourth webinar in our Generation Chemical Partnership series, which is titled Preconception Matters 2, EDC Impacts on Reproductive Health and Development. Our moderator today is Dr. Janet Hall, Senior Investigator at NIH and Chair of the Clinical Research Branch and Clinical Director of the National Institute of Environmental Health Sciences. We will leave time following the presentation for a brief Q&A session and panel discussion. You may type in questions through the Q&A feature available on the menu bar at the top of your window at any point during the presentation. After the presentation, our moderator will read out questions for our speakers to respond to. We will get to as many comments and questions as we can during the Q&A period, and we'll follow up on unanswered questions as we can. For those of you who have called in on the phone, we have posted today's slides to accompany our webinar on the website. You can download these by going to healthandenvironment.org. Please scroll to the bottom of the page and select today's webinar. On the webinar page is a link to the slides. Everyone on our webinar right now is muted with the exception of our moderator and our speakers. The webinar is scheduled to last for 70 minutes and is being recorded for our call and webinar archive. With that, I would like to introduce Dr. Hall. Dr. Hall is a senior investigator at NIH and chair of the Clinical Research Branch and clinical director of the National Institute of Environmental Health Sciences. She completed her undergraduate, graduate medical and internal medicine training at McMaster University in Ontario, Canada, and her fellowship in endocrinology and metabolism at Massachusetts General Hospital. Dr. Hall spent much of her professional life at MGH with consistent research funding from NIH and rising to the rank of professor of medicine at Harvard Medical School before taking her current position at NIH. She's an international known clinician and clinical investigator with over 200 peer-reviewed publications, as well as major chapters in the leading textbooks of medicine and reproductive endocrinology. Her research focuses on neuroendocrine and ovarian integration and control of reproductive systems in health and disease, including the effects of endocrine disrupting chemicals, physicians on the reproductive health system, Dr. Hall is a member of the Association of American Physicians and is past president of the Endocrine Society and of Women in Endocrinology. She has been associate editor of both the Journal of Endocrinology and Metabolism and Endocrine Reviews, and is a recipient of the Sidney Ingbar Award for Distinguished Service to the Endocrine Society. She currently serves on the Endocrine Society's Advisory Committee on EDCs. And now I'd like to turn it over to you, Dr. Hall.
Wonderful. Thank you so much, Hannah. With a session that begins at 1 o'clock Eastern Standard Time, 1 o'clock p.m. Eastern Standard Time, but 11 a.m. Pacific Time, I now understand why the Australians, who are always in different time zones from everybody else, always use g'day as their greeting. So g'day and welcome to the fourth in our collaborative on health and the environment series, Generation Chemicals, How Environmental Chemicals Are Affecting Reproductive Health and Development. If you didn't have the opportunity to view the previous sessions live, I would really like to encourage you to do the archived presentation. It's just a wonderful opportunity to hear from the experts in this field. This is a field that actually has grown from really the very, I don't know, it's really a nascent field about a decade ago. But over that time, it has gone from a very small number of investigators who've really been interested in the reproductive implications of things in our environment to one that has attracted really an impressive number of mature, high-class scientists, as well as early career investigators who are making remarkable strides. Today we're going to be, as I mentioned, focusing on preconception exposures. Subsequent sessions are going to be focused on fetal and maternal health. So this again is a series that is going to really expand and cover the breadth of reproductive outcomes. So As an endocrinologist, actually, my first introduction to the importance of preconception exposures actually came in the form of the profound negative effect of poorly controlled diabetes and that effect of the exposure to high glucose levels on fetal outcomes and longer-term outcomes in children. But of course, since that time, more recent research suggests that there are certain maternal and paternal chemical exposures both prior to and at the time of conception that impact reproductive health and have transgenerational effects. So endocrine disrupting chemicals, or EDCs, are ubiquitous in our environment. They're found in food packaging material, water, air, personal care products, furniture, clothing, and more. Maternal and preconception exposures to individual mixtures of endocrine disrupting chemicals like phthalates, parabens, and phenols that are going to be discussed today have been linked to multiple adverse reproductive and birth effects, reproductive health and birth effects, including early onset of puberty, reproductive cancers, low birth weight, and preterm birth. So our speakers today are going to be discussing the important studies in which they have been involved that are adding critical data to this really important area. I'm going to introduce all three of our speakers now before we turn to our first speaker, Kim Harley. But Kim Harley is a reproductive epidemiology and a faculty member in public health at UC Berkeley. Her research focuses on endocrine disrupting chemicals and women's health, including associations with fertility, birth outcomes, timing of puberty, and obesity. She's the associate director of the Chamacos, and I'm sorry if I murdered that study, a longitudinal cohort study examining environmental exposures to immigrant farm worker women and their children in California. Dr. Harley is also a principal investigator of several youth empowerment projects that train young people to identify environmental justice problems in their communities and conduct public health research to address them, including the Hermosa study of young girls' exposures to chemicals in makeup. Her latest research project investigates chemicals of concern in cosmetics and personal care products marketed toward minority women. She's also the principal investigator of an intervention to reduce women's chemical exposures through their household cleaning products. Dr. Harley is an incredibly well-published investigator with more than 100 peer-reviewed papers on health effects and exposure patterns of phthalates, parabens, PPA, flame retardants, pesticides, and other endocrine disruptors in our homes and daily lives. So welcome, Kim, but give me a second. So the next, our next speaker that I wanted to introduce is Carmen Masurlian. Carmen is an assistant professor of environmental, reproductive, perinatal, and pediatric epidemiology at the Harvard T.H. Chan School of Public Health, and as well as the director of the Scientific Early Life Environmental Health Study program, which is a SEED program. Her research is focused on examining the extent to which environmental exposures affect a couple's ability to achieve conception, to maintain pregnancy, and to deliver healthy offspring. Dr. Masurlian investigates paternal and maternal exposures to phthalates, phenols, PFAS, and other emerging chemicals and their mixtures on ovarian reserve, time to pregnancy, pregnancy loss, preterm birth, birth weight, placental parameters, and child development outcomes. She applies cutting-edge epidemiologic methods to generate evidence-based knowledge of the effects of environmental chemicals on fertility, pregnancy, and child health outcomes. Her ultimate goal is to use this evidence to inform clinical practice and improve health for mothers, fathers, and children. Also with us today, as a very special guest, is Yu Zhang, who is a doctoral student in environmental and reproductive epidemiology at the Harvard T.H. Chan School of Public Health. She received her bachelor's degree from Peking University in China. Her research under Dr. Masurlian's mentorship aims to understand how endocrine disrupting chemicals, including phenols, phthalates, and polyfluoroalkyl substances influence perinatal outcomes, with a focus on identifying critical periods of vulnerability and underlying pathways. She examines preconception chemical exposure in both parents in relation to pregnancy loss, preterm birth, and birth weight, and applies advanced statistical methods to study the joint effect of chemical mixtures on couple-based outcomes. She also explores environmental exposures during the prenatal window on adverse birth outcomes. So first of all, thank you to all three of you for agreeing to be part of this series. And without further ado, I would love to introduce you to Kim Harley as our first presenter, speaking about the association of phthalates, parabens, and phenols in personal care products with pubertal timing in girls and boys, an area that is really important in our current environment. Kim.
Thank you, Janet. Can people see me and hear me or see my slides?
We're all good.
Great. So I'm going to jump right in here. Let's see. First, let me see if I can make my slides advance. There we go. So I'm talking about endocrine disruptors today that are found in personal care products. And when we talk about personal care products, these are things that we all use. So we're not just talking about cosmetics. We're talking about soap and shampoo, deodorant, toothpaste, things that are really ubiquitous in our everyday lives. But we do know that women tend to use more personal care products than men. One small study found that the average woman uses 12 personal care products a day, and teenage girls use even more. They use about 17. So we're concerned about exposures to women and also to girls during this time of reproductive development around puberty and in adolescence. There's also some evidence that more than 12,000 chemicals are used in cosmetics and personal care products. So this is a lot of potential for exposure because we know that these chemicals have the potential to enter our bodies, whether it's through inhalation, we're breathing them in, through ingestion, or through dermal absorption. So specifically today, I'm going to be talking about some suspected endocrine disruptors in personal care products, and I'll talk about 4 classes of chemicals that people are probably familiar with. We'll talk about phthalates. These are the low molecular weight phthalates. The high molecular weight phthalates are used more in plastics. Low molecular weight phthalates like diethyl phthalate or the dibutyl phthalates have been used in personal care products. Diethyl phthalate is the most widely used, with the one with the highest levels of exposure, and that has been used in fragrance. We're also looking at parabens. So parabens are preservatives that are used in a lot of makeup. There are many different kinds of parabens. The most common ones are methyl and propylparaben, which we'll be talking about, but there are many others that are also used as preservatives. Triclosan is an antibacterial agent that was very widely used in antibacterial soap and was in some toothpaste. Triclosan is actually no longer in these products. The FDA ordered triclosan to be removed from antibacterial soap in 2016, and it was voluntarily removed from toothpaste in 2019. So it's not so much of a current exposure, but was an exposure when we did our study. And then the last chemical that we'll be looking at is benzophenone 3, BP3, also known as oxybenzone, which is a sunscreen agent. And one of the things about these chemicals, these 4 classes of chemicals that I'm showing you here, is we really know that their primary source of exposure is through personal care products. And just to illustrate that, I'll show you some data from a study we did a few years ago where we had teenage girls and we took urine samples from them. I should mention the 4 classes of chemicals we're talking about are metabolized in the body and excreted as in urine. And so we can take urine samples and find the amount of these chemicals in people's bodies. And these are some teenage girls. We brought them in and took a urine sample from them and asked them if they'd used, in this case, sunscreen in the past 24 hours. And you can see significantly higher levels of BP3, for example, in the girls who said that they'd used sunscreen. And here's just another example looking at propylparaben. You can see much higher levels of propylparaben in the urine of girls who said that they'd used foundation, blush, or mascara in the last 24 hours. So today we're going to be talking specifically about timing of puberty. And people are probably familiar with this because timing of puberty has gotten a lot of press in the last several years. There's quite a bit of evidence that girls are now entering puberty earlier in the United States. And there's now been several studies to show that this is also probably true in Europe and probably around the world. This is a meta-analysis that was published about a year ago, showing that over the last 40 years, we've seen that the onset of breast development has decreased by about 1 year. And I do want to mention that we know that obesity, childhood obesity, is a major factor in this trend towards earlier puberty that we're seeing, without a doubt. But the question is, are some of these endocrine disrupting chemicals that we're exposed to in our daily lives also contributing to this phenomenon? So just to back up for a minute, how do we measure onset of puberty or pubertal timing? The answer is, the gold standard anyway, is to use clinical Tanner staging. So this is a clinical exam where you examine the breast and genital development of the child as they're advancing through puberty. Specifically, we're looking at onset of breast development. So the very first breast development, known as theliarchy, is the sign of onset of puberty in girls. We're also interested in pubic hair development, and menarche, of course, is an important pubertal milestone, but it's not the onset or the beginning of puberty. So in girls, breast development is the primary marker, but these other milestones are also important to look at. In boys, we know that puberty has started with the increased testicular volume and genital development. And again, we also look at pubic hair development in boys. And I do just want to mention that this trend that we're seeing globally towards earlier puberty in girls is quite well established at this point. But whether that trend is also happening in boys is a lot less clear. We don't really know if boys are entering puberty earlier or not. There's been some conflicting research on that. So if we're looking specifically at these chemicals that we're talking about, those endocrine disruptors, the phthalates and the phenols that are in personal care products, I wanted to just take a minute to look at the animal research on whether they're associated with timing of puberty. And there's only a small number of studies looking at these chemicals in animals, but we do see in some studies that those low molecular weight phthalates have been associated with earlier puberty in female rats and later puberty in male rats. Similarly, same thing with parabens. Again, this is a very limited number of studies. And triclosan has been shown to be associated with earlier puberty in rats. Now, there's some differences in these studies. Some of them are prenatal exposure. Some of them are peripubertal exposure. And I've just included one graph here on the side where you can see earlier onset of vaginal opening, which is onset of puberty in female rats with higher levels of triclosan. So there is a small amount of literature in rats. There's an even smaller amount of literature in humans in the epidemiology literature. And many of those studies are cross-sectional. To my knowledge, there's only 3 studies that are longitudinal that are able to look at earlier life exposure to these chemicals and whether that's associated with timing of puberty. And some of those studies have looked at prenatal and some of them have looked at childhood exposure. Which brings us to the research question of our study, which is specifically, is timing of puberty in children associated with personal care product chemical exposure at 2 critical time points? So we're looking at levels of these chemicals in maternal urine during pregnancy as a marker of in utero exposure, and also metabolites of these chemicals in child urine at age 9, which is pre-puberty or sort of peri-puberty exposure. This research was conducted in the Chamacos longitudinal birth cohort study. This is a study that we've been doing for 20 years now in Northern California here. We enrolled about 600 pregnant moms in 1999 and 2000, attending prenatal care clinics, serving the Spanish-speaking, primarily farm worker population in the Salinas Valley, which is an agricultural region south of here. So we enrolled the moms in the year 2000. They are mostly Spanish speaking. They were mostly born in Mexico. And then we saw those moms twice during pregnancy, took 2 urine samples, which we were able to measure the exposure in. And then we've seen the children at multiple time points, and we were just finishing our 18-year visit now. Between the time that the children were 9 and 12 3/4, we saw them every 9 months. And during that time, we conducted clinical Tanner staging. So we have up to 6 visits where we were able to measure what stage of puberty they were in. And we had 338 children who were still in our study at that time point. We also took a urine sample from the children at age 9. So that's where we're getting our exposure measures. 2 urine samples averaged during pregnancy, 1 urine sample from the child at age 9. And then those samples were sent to CDC for measurement of phthalates, phenol, and phenols. So here's what we found with our associations with timing of puberty. And first, I'm going to take a minute to orient you to this graph because there's a lot here. First, we're looking at the association of prenatal urinary metabolites with puberty in girls. And so what we see here, we used accelerated failure time models to get a time ratio, so a shift in time associated with each doubling of exposure to these chemicals. So we have MEP, MBP, and MIBP. Those are the three low molecular weight phthalates. TCS is triclosan, BP3, and then the two parabens. And what you see looking here is most of these cross zero. In fact, they all cross zero. I will draw your attention to MEP. We see a shift. It crosses zero, so it's not statistically significant. But we do see a trend towards about a -1, so about a 1-month shift in earlier puberty, earlier breast development among children whose moms had higher levels of MEP in their urine. When we look at pubic hair development, we actually see a statistically significant decrease in timing of puberty for pubic hair development, again, with moms who had, for each doubling of MEP, which is the metabolite of diethyl phthalate, the one that's found in fragrances, I should specify. When we looked at timing of menarche, what we saw in this one, in contrast, was a little more than a 1-month decrease for each doubling of maternal triclosan. And you can see on the side here what we've adjusted for. So this is looking at these exposures as a continuous variable. And my next slide, I'm going to zoom in a little bit on those two. So we saw an association, as I mentioned, with MEP, the metabolite of diethyl phthalate. And so here, let's look at that specifically. Instead of looking at all of the moms with that as a continuous exposure, now we've divided the moms into 4 groups. So Q4 is the highest quartile of exposure, and Q1 is the lowest quartile as the reference group. And you can see here that when we compare the children, the daughters of the moms in the highest quartile of exposure, they entered breast development, thelarche, about 6 months earlier compared to those whose moms were in the lowest levels of exposure. Similarly, when we look at pubarche, or timing of pubic hair development, we see, again, almost a 6-month decrease in the age that they entered puberty compared to those whose moms were in the lowest levels of exposure. Now I'm going to look at triclosan the same way. So remember, triclosan is that antibacterial agent that used to be in toothpaste until very recently. And again, when we compare the daughters whose moms had the highest levels during pregnancy to those with the lowest, we see a 4-month decrease, 4-month earlier onset of menarche with triclosan exposure. And this is somewhat consistent with what was seen in the animal studies, although there's really only very few. Okay, so this is prenatal exposure. Now I'm going to shift and look at what we saw with childhood exposure, which, remember, was measured when the children were 9 years of age, and which was just as they were starting puberty. Unfortunately, we don't have phthalate measurements in the 9-year urine, so we're only able to look at the triclosan, the BP3, and those two parabens. But again, we see that girls who had higher levels of methylparaben in this case had earlier timing of onset of breast development. And when we look at pubic hair development, we see, again, earlier pubic hair development with each increase, 2-fold increase in methylparaben, borderline with propylparaben. And looking at menarche, again, we also see that pattern. So what you're seeing here across all 3 measures of pubertal timing in girls is a trend, in some cases significant, some cases not, with methylparaben and maybe propylparaben towards earlier timing of puberty. And again, I'm going to zoom in on the methylparaben and again divide them into 4 groups. So looking at the same thing slightly differently, if you look at the girls who were in the highest quartile of methylparaben at age 9, nothing really with breast development, but with pubic hair development and with menarche, you can see that they are entering pubarche and menarche about 6 months earlier than their peers who had very low levels of exposure. Now, I do want to put a caveat on this, that this is exposure at age 9, and it's happening as puberty is happening. So it's possible, I could imagine a scenario, for example, where you have girls that are more advanced who are maybe wearing more makeup and having more exposure. So when I showed you the prenatal exposure, I could feel really confident that that was exposure that was happening before puberty. In this case, there's maybe a possibility of reverse causality that I just want to put out there as a caveat. So that was girls. I'll tell you very quickly about boys because we didn't really find anything with boys. If you look here at the prenatal exposure, they all cross the zero line. We found no association with boys. And when we look at the boys' exposure at age 9, they pretty much all cross zero. There is some evidence that propylparaben might be associated with earlier genital development, but that's the only association we see with boys. So in summary with this finding, we found that several chemicals in personal care products were associated with earlier puberty in girls, but not with boys. And that we found associations with both prenatal and peripubertal exposures, specifically in the prenatal exposures, the highest maternal exposure to diethyl phthalates was associated with a 6-month earlier onset of pubic hair development, and the highest maternal exposure to triclosan was associated with 4 months earlier menarche. And when you look at the peripubertal, we see that the highest childhood exposure to methyl and propylparabens was associated with about 6 months earlier of multiple measures in girls. We do have some reasons for caution. I just want to point out that this is really one of the first study to examine prenatal exposures to some of these personal care product studies. There's been one other study that looked at phthalates and did similarly find an association with diethyl phthalate metabolites and earlier puberty, but no other studies have looked at the phenols like the parabens, the triclosan, and the BP3. So obviously this needs to be replicated in other studies. And also, this is a very specific population. This is a population of low-income rural Latinas living in California. So another reason for replication being necessary. We are able to measure levels of these chemicals in the body using urinary biomarkers, which is great. But I think we all need to remember that these are short-term exposures. And so when we're measuring urine levels of these chemicals, it's really only reflecting exposure in the last 24 to 48 hours. Now, a lot of people use the same personal care products over and over again for every day. And so there is some consistency in these measures, but that is definitely a weakness. And also, as I mentioned, the associations with childhood exposures might actually reflect reversed causation. So just to speak briefly about some of the policy implications. I think my slides might be out of order. Yeah, there we go. So we know that the chemicals in personal care products are getting into our bodies. And now this study provides evidence that these suspected endocrine disrupting chemicals may be impacting pubertal timing. And I just want to point out that there are a lot of chemicals in personal care products. The FDA has authority over cosmetics, but they have very limited authority over personal care products and cosmetics in general. They don't have the ability to recall products or to demand testing, pre-market testing. We are seeing some regulation of cosmetics. It's slowly starting to change. For example, California passed last fall a California Toxic Cosmetics Free Act, which will ban, I believe, 12 chemicals. Only one of them is one of the chemicals we looked at, though. So there's some chemicals here that are not affected by that. I just want to end with 2 more slides just to hopefully show you that switching your products can reduce exposure. So on a personal level, there are ways that we can reduce our exposure to these chemicals. This is a study we did a few years ago where we enrolled 100 teenage girls in our community in Salinas, California. And we collected a urine sample from them. And then we asked them not to use their regular personal care products for 3 days. You can see a picture here. This is our beauty bar. We let them choose low chemical products that said on their labels that they didn't contain phthalates, parabens, or any of the chemicals that we were concerned about. They came back 3 days later after not using the regular products and gave us another urine sample. And you can see that after 3 days of not using the regular products and using lower chemical products, we saw a 28% decrease in diethyl phthalate metabolites, a 35% decrease in triclosan, a 36% decrease in oxybenzone, BP3, and a 44% decrease in both methyl and propylparabens. So just in 3 days of changing their personal care products, we were able to see really significant changes in their exposure. So I'm going to end on that. and just say thank you to our funders and our research team, and particularly to the families in our Chamaco study.
Dr. Harley, thank you so much. That amazing. Thank you for sharing the results of your studies, as well as your thoughts about regulation and where we might go with this. I know there's going to be a lot of questions to follow, but really appreciate that. Dr. Moussolian and Dr. Zhang, I think you're on. But you need to unmute yourself.
Sorry about that. Well, I'll redo my introduction. Thank you, Dr. Hall. I realize you're Canadian as well. So hello. Good day to everyone. My name is Carmen Moussolian. We will be presenting jointly with my doctoral student, Yu Zhang, from the Harvard Chan School of Public Health. We're really excited to be here today, and we're really happy to have Kim Harley do the earlier presentation because she covered a lot of elements within our presentation that are really relevant and important, in particular telling us about the sources of exposure, which are important determinants of the types of chemical mixtures that we see in women and men. In addition, you talked about your intervention study, which leads into our work throughout this process of understanding how these chemicals impact reproductive health. And we're now working on an intervention study among couples. So thank you for your presentation, Dr. Harley. So the title of our talk is Maternal and Paternal Preconception, Endocrine Disrupting Chemical Exposure and Birth Outcomes. And with that, I'm going to switch slides. So I'll tell you a little bit about the outline of our presentation. So I will do the background and talk about the study design. Yu Zhang will talk about the methods and really get into the mixtures analysis methods that we use to undertake a more complex assessment of phenols and phthalates. And Yu Zhang has become an expert in this method, so I will leave her to explain that with more detail. And then we'll present results on work that is under review presently on preterm birth and birth weight. So we looked at two different important adverse perinatal outcomes, and we'll present results on mixtures analysis in that. And then I will conclude with some of the summary of the findings that kind of combine the single chemical analysis that was previously published by our group, as well as the chemical mixtures analysis to sort of give you a comprehensive picture of which chemicals really matter and which windows of exposure are important. So I'm going to switch on to the next slide. Before that, I would like to just introduce, I think my introduction was very nice, so Dr. Hall, thank you, but I wanted to introduce our SEED program. The SEED program, I'm the PI of that program, but I have an amazing group of colleagues and students and postdocs, and I encourage you to look us up and identify yourselves if you're interested in the kind of work that we do. So thank you. So we know that healthy reproduction requires complex hormonal processes to work in synchrony. Given their potential to disrupt hormonal homeostasis, EDCs may alter critical pathways required to achieve conception, maintain pregnancy, and deliver healthy offspring. And there's ample toxicological evidence that shows that some EDCs are reproductive, neurological, and epigenetic toxicants. And many are also teratogenic and obesogenic. There's also an accumulating body of epidemiological evidence that links these EDCs to adverse reproductive and developmental outcomes across the reproductive life course. We heard about pubertal timing earlier with Kim, and that's an important reproductive outcome that needs further study as well. But we're focusing more on birth outcomes. And in particular, we want to convey the importance that pregnancy and birth and fertility is a couple-based outcome, and they're couple-based exposures. And so we really need to look at both the male and the female partner in order to really understand the comprehensive complex interplay between fathers and mothers and how reproductive success happens. And our knowledge in this area is really still in its infancy, and we're still trying to understand the impact that fathers have on reproductive success. But something that's really important is the timing of exposure. And the timing of exposure is really a critical determinant of disease susceptibility. Different periods in the developmental life course are more or less susceptible to the effects of EDCs. For example, embryogenesis is governed by complex genetic and epigenetic mechanisms, and exposure during this period, or perhaps even before conception, may be the critical window during which environmental perturbations may exert their adverse effects and increase the risk of specific adverse outcomes, such as preterm birth, birth weight. Moreover, depending on how we design a study, including the timing of when we measure exposure and outcome will have an important impact on what we find. So if we don't measure exposure in the preconception period, we won't be able to identify the impact of that preconception exposure on things like oogenesis, spermatogenesis, uterine receptivity, and overall health. And the preconception window is really important because it not only sets up the parameters and the determinants of the biology of reproduction, but it also impacts the general health of a woman or the general health of a man, which can impact how they reproduce. And so this has been sort of the interest of ours. Well, I'm going to just keep going. So let me switch slides because I'm talking over the first slide. Okay, so while there is substantial evidence linking phthalates to reproductive endpoints in animals, less is really known about the effects in humans. And most studies have measured exposure in the prenatal window, and few studies have really looked at the preconception exposure. And there's fairly robust data on prenatal phthalate exposure in preterm birth. And several studies by Ferguson and Meeker and others have shown that phthalate exposure, in particular DHP or diethyl hexyl phthalate, increases the odds of preterm birth by about 30 to 70%. And phthalates have also been associated somewhat inconsistently with things like preeclampsia, reduced birth weight, and other pregnancy adverse outcomes. But differences in study design and measurement likely contributed to the differences in findings. And looking at conception-specific exposure, our earlier work using data from the EARTH study, PI Russ Hauser, found that diethyl hexyl phthalate was associated with other adverse reproductive outcomes, including a reduced oocyte yield, fewer mature oocytes, and reduced clinical and live birth rates among couples in a fertility setting. And now, since we've really examined sort of the individual chemical models, so single chemical models at a time, so looking at individual phthalates or individual phenols such as BPA and looking at adverse reproductive outcomes, we really need to understand the complex mixtures. And so beyond the single chemical problem, there's real world exposure scenarios that we're not able to tap into without more complex methods. And this has really inspired us in the last year to adapt and adopt certain types of methodological tools to be able to better understand the complexity of real world scenarios of complex mixtures. And something in addition to the timing of exposure, looking at the preconception window, is really the fact that chemicals in their mixtures are not just endocrine disruptors. They're also epigenetic toxins. They alter epigenetic regulation. They also impact imprinting of the genome through early embryo and fetal development. We also know that these chemicals are immunotoxic and they impact things like inflammation and immune function. And they're also impacting metabolism. Our metabolism is impacted by some of these chemicals, and we know that some of them are obesogenic. And all of these processes are really important for successful reproduction. And so looking at the complexity of mixtures allows us to look at multiple pathways and mechanisms, or at least give us some speculation, some information that could be used to understand sort of the mechanisms. And our hypothesis in this work is really that this preconception window, these processes that happen that set up reproduction either through the endometrial lining, the uterine receptivity, the way the oocyte gets signaled out, the way spermatogenesis happens, all these really early reproductive steps or molecular steps are really important to determine the outcome of the pregnancy. And so we decided that looking at the preconception window would help us inform sort of this hypothesis that these chemicals act early on in the pregnancy to impact oocytes and spermatogenesis, including things like uterine receptivity. And then through those disrupted molecular processes, we can have a disrupted pregnancy through disrupted fertilization, disrupted implantation, and potentially disrupted embryogenesis. And this is supported really quite substantially from animal studies and experimental studies, but hasn't really been looked at epidemiologically per se. And so we hypothesize that fathers and mothers' exposure before pregnancy through these pathways could impact birth weight and preterm birth. And as such, the objective of this work was to determine the extent to which maternal and paternal preconception urinary concentrations of phenols and phthalate mixtures were associated with adverse birth outcomes. And to answer this question, we used the Environment and Reproductive Health Study. Again, the PI is Russ Hauser, and that's a prospective preconception cohort that took place at the Massachusetts General Hospital Fertility Clinic from 2005 to about 2018. And in the study, we recruited men and women couples. Sometimes the women would come with their male partner, sometimes the woman would come independently. and occasionally a man would come without his female partner. And we recruited couples as they attempted conception. And the model for the study really allows us to tap into very early reproductive endpoints, including fertilization, implantation, and pregnancy loss, early pregnancy loss, live birth, as well as preterm birth and birth weight. And so in order to answer the questions about the preconception period, we averaged urine that we collected from the couples in the time of entry to the study all the way until the conception cycle, so the cycle of conception of the index pregnancy. And we averaged exposure over several months to be able to collect a fairly well, there's always measurement error with all exposures that are short-lived, but a little bit more information in terms of their total or overall exposure in the window before conception. And we did this for men and for women. So we had urine samples collected at each cycle for men and urine samples collected at each cycle for women. And we averaged this window to be able to identify overall exposure for men and women. And with that, we were, let me just switch slides. We were also able to look at, through the Centers for Disease Control, send the urine samples out. And Antonia Califat's laboratory measured 11 different phthalate metabolites and a host of phenols as well, including BPA and parabens. We also measured triclosan and benzophenone 3, but they do not form part of this particular analysis at this time. And we have published previously looking at the individual chemicals, but we haven't really published that much in terms of the birth outcomes with respect to the mixtures of chemicals. And this is the point of this particular presentation. So what do we look at for outcomes? We use the medical records of the women that gave birth at the we use, someone's not muted, someone with a little child, I get interrupted too. Okay, so as we followed the couples along, we waited for their birth outcomes and we measured and followed them up through pregnancy. And then we obtained their medical records and the delivery records to identify the birth weight, the gestational age, and we looked at preterm births, so births before 37 weeks gestation is one of the outcomes. So, well, we are not going to present too many details on the single chemical analysis because that was previously published by Mustielis et al. 2020 and Yang and Zhang et al. 2020 as well. So you could look those papers up, but I will pass on the baton to my colleague and doctoral student, Yu Zhang, to take over the presentation from this point. Yu Zhang can unmute herself. She will go into more about the analysis for the mixtures methods that we used. So thank you for that. Oh, sorry about that, Yu. And I'll advance.
Thank you, Carmen.
I'll advance the slide.
Thank you. Thank you. It's my honor to be here and present the methods. Yes. So we assessed maternal and paternal preconception mixtures respectively. And we also to look at couples' joint exposure on the outcome. We fit both maternal and paternal mixture into one model. And we utilized two complementary mixture methods, the principal component analysis, the PCA, and Bayesian kernel machine regression, the BCMR, to evaluate the association between maternal, paternal, and the couple's joint preconception phenols and phthalates mixture in relation to birth outcomes. Could you go to the next slide, Carmen? Next slide. The next slide briefly introduces the two mixture methods. PCA is an exposure dimension reduction method. It reduced the mixture into uncorrelated groups based on the correlation structure. And the method is very useful to know the real-life exposure pattern and potential shared sources of groups within the mixture. We then fit regression models to the PCA-derived groups to examine the associations between the PCA-derived mixture groups and birth outcomes. We utilized these PCA methods for maternal and paternal windows respectively. Because we cannot know the association between individual biomarker within the mixture and birth outcomes using PCA, so we complement PCA with BCMR, which is a very flexible method to evaluate mixture and birth outcomes, taking into consideration of correlation, interaction, and nonlinear relationships. And BCMR can inform us of the univariate associations in the context of mixture, the interactions within the mixture, and it can also shows us the cumulative effect of the mixture on the birth outcomes. Using the hierarchical variable selection, BCAMR can evaluate the relative importance of groups which we assign in fitting the model. And the relative importance is demonstrated by posterior inclusion probability. And the PCA informed the groups we assigned in the hierarchical variable selection for both maternal or paternal preconception windows. And in the couple-based model, where we entered both maternal and paternal exposures simultaneously, we divided the total mixture into two groups, the maternal group and the paternal group. The aim was to compare the relative importance of mixture from the maternal preconception window and the paternal preconception window. Next slide is on the covariates. The covariates were selected a priori as potential confounders based on substantive knowledge using causal diagrams. And the covariates in the maternal model included maternal age, BMI, education, smoking status, race, and mode of conception. And the paternal model included the covariates in the maternal model and also the corresponding paternal covariates. And the model for couples included both maternal and paternal covariates. And then I'll show the study results for preterm birth and birth weight. So first, about our population. In our studies, we included 384 females and 211 males, which constituted 203 couples. And for the females, they had a mean age of 35, and 85% of them were nulliparous at study entry. And the males had a mean age of 36, and 27% of them had a male factor as their infertility factor. And both males and females were mostly white, highly educated, and non-smokers. And then I'll show the mixture results for preterm birth. And in the next slide, the table shows the risk ratio of preterm birth for every unit increase in PCA factor scores. And we derived the same 4 factors from PCA for both maternal and paternal mixtures, which we named as DHP and BPA factor, paraben factor, high molecular weight phthalate factor, and low molecular weight phthalate factor. And we then fit these 4 PCA-derived factors into Poisson regression to get the risk ratio of preterm birth. And we observed positive associations for maternal and paternal DHP and BPA factor scores in preterm birth risk. And there's also some suggestive association between paternal paraben factor and preterm birth risk. And then on the next slide, the BKMR derived figure depicts, on the next slide, the BKMR derived figure depicts the association between a given biomarker concentration and preterm birth estimate when holding all other biomarkers at their median concentrations. And this figure is for the maternal preconception window. The x-axis is the natural log transformed biomarker concentrations, and the y-axis is the preterm birth estimates. We can see a clear increasing trend between maternal BPA concentration and preterm birth when holding the rest of the biomarkers at their median concentrations. And the next slide, the figure in the next slide shows the univariate associations for paternal preconception window. We observed some suggestive positive associations for the 4 DHP metabolites, although they were very imprecise. And we showed the cumulative effect of the mixture on the next slide. In this figure, the x-axis is the quantiles of the total mixture concentrations for maternal, paternal, and couples preconception total mixture concentrations. And the y-axis is the preterm birth estimate. And we hold the median concentration as the reference group. And we observed increasing trends of preterm birth estimates across quantiles of maternal and couples total preconception mixture concentrations. And on the next slide, as for the posterior inclusion probability, the maternal and paternal BCAMR model confirmed that the DHP and BPA factor was important on the association with preterm birth among the identified 4 PCA factors. And the couple-based model showed very similar importance of maternal mixture group and paternal mixture group on the outcome. So in summary of the BKMR results for preterm birth, maternal preconception BPA and paternal preconception DHP metabolites were associated with higher preterm birth estimates when holding all other biomarker concentrations at their median levels. We observed higher preterm birth estimates across quantiles of maternal and couples total mixture concentrations. And then I'll present the results for birth weight. And this table shows the change in birth weight for every unit increase in PCA factor scores. And again, we derived the same 4 factors from PCA for both maternal and paternal models. And the paternal preconception DHP and BPA factor and the low molecular weight phthalate factor seem to be associated with decreased birth weight. And when we look at the BKMR results, and this figure shows the univariate associations for maternal preconception window. We observed a decreasing trend of birth weight across concentrations of maternal preconception BPA when holding the rest of the biomarker concentrations at their median levels. And the next slide shows the paternal univariate association. We observed a negative association between MBP and birth weight when holding the rest of the biomarker concentrations at their medians. And the MBP is included in the low molecular weight phthalates. This slide shows the cumulative effect of the mixture on birth weight. We observed clear decreasing trends of birth weight across quantiles of maternal, paternal, and couples total preconception mixture concentrations. And for the posterior inclusion probability, the paternal BKMR model showed high inclusion probability of the DHP and BPA factor, and also low molecular weight phthalate factor, which were consistent with our PCA results. And the couple-based model showed that paternal mixture group had a higher inclusion probability than maternal mixture group, which may suggest that paternal preconception exposure might play a more important role on birth weight than maternal preconception exposure. So summary for the BKMR results for birth weight, we observed maternal preconception BPA exposure and paternal preconception MBP exposure were associated with reduced birth weight when holding all other biomarkers at their median concentrations. We observed clear decreasing trends of birth weight across quantiles of maternal, paternal, and couples total mixture concentrations. So these are all our results, and now I hand it over to Carmen to talk more about the conclusions and implications.
Sorry about that. Technologically challenged here today with switching around between 2 slides. Thank you very much, Yu Zhang. And I have to say, Yu Zhang's really done an amazing job learning this complex method. And she's presenting from Beijing, China today, which I think it's around 2 in the morning for Yu Zhang. So thank you so much. You did a wonderful job. I'm very proud of you. So in summary, that's a lot of information, a lot of different windows of exposure, a lot of different methods. We want to be able to now help you sort of digest these findings and summarize them a little bit better for you. So this is a basic summary of the findings that we identified through this work on preterm birth as the outcome. And there's some consistency with respect to the different models, looking at mixtures, as well as the single chemical analyses that we published on previously. And in the single chemical analyses papers that we published, we did identify that both maternal and paternal exposure to DHP and BPA increased preterm birth risk. And in the mixtures analysis using PCA as well as BKMR, we again found that both maternal and paternal exposure to diethylhexylphthalate and bisphenol A increased the risk of preterm birth. In addition to that, we found that father's paraben exposure might be associated with an increased risk of preterm birth. And we did identify similarly methylparaben in our single chemical analysis to be associated with a higher risk of preterm birth. Importantly, in this work, the BKMR analysis allowed us to look at the complex mixture. So just to elaborate a little bit more on that, the complex mixture actually increases the totality of all those individual biomarkers by one quantile at a time. And so it allows us to look at the cumulative effect of the exposure among mothers and fathers independently on preterm birth, and then as well as among couples on preterm birth. And here we found, I think, the most relevant finding, which is that, that the total mixture among couples increased the risk of preterm birth. And when we look at the probability of inclusion, the PIP factor that Yu Zhang described, it's a component of BKMR. It let us actually see the fact that the father's and the mother's exposure actually contributed equally to the adverse outcome. And so this suggests that it's not just the mother's exposure that might be important here, but also the father's. And we don't really understand the mechanisms per se. However, some of the animal studies that we've looked at have shown independent chemicals such as DHP through exposure through fathers could impact, if you look at papers by published in Human Reproduction by Rick Pilsner at UMass Lowell, he showed, or sorry, UMass Amherst, he showed that some of the epigenetic mechanisms associated with spermatogenesis and DNA methylation on sperm epigenome actually altered things like embryo development, embryogenesis, and development. There were genes that were enriched in those parameters for growth of the embryo. And so this leads us to be concerned about father's exposure before conception, as well as mother's exposure before conception. Okay, so what did we find in conclusion for preterm birth risk? So we found that mothers' exposure to DHP and BPA as a PCA factor increased the risk of preterm birth. And so did BPA independently of all the other chemicals. So when we looked at those chemicals, all the other chemicals in the mixture at their median concentration, increased levels of BPA were associated with higher risk of preterm birth. And again, the total mixture among mothers also increased the risk of preterm birth. In fathers, we found similarly that the DHP and BPA factor increased the risk of preterm birth independently of other chemicals in the mixture. And we also found that paraben exposure might be associated with a higher risk of preterm birth. One particular caution in this work is that our sample size is relatively small. We had about 203 couples and a little bit over 217 men. And so some of our precision is limited because of the sample size, but I think the trend is important, as well as the consistency in our results from our studies, as well as other studies that have shown similar types of metabolites or biomarkers that were associated with a higher risk of preterm birth. Now moving on to birth weight. This is another figure or table that shows us the totality of the findings. And again, we've identified that the DHP factor among fathers, the DHP-BPA factor among fathers, was associated with reduced birth weight through the PCR models. And again, this was really consistent with our single chemical analysis that found that it was fathers' exposure to DHP and not mothers that contributed the most to a lower birth weight. And we found this as well through this PC. Oh, this is supposed to say PCA, by the way. Yu Zhang, remember last night we forgot to change it. Anyways, I made a mistake. I was thinking about COVID testing and wrote PCR. Apologies. So the BKMR models, we found that BPA in mothers, independently of the other chemicals in the mixture, was associated with reduced birth weight. Again, fathers MBP, monobutyl phthalate, was associated with lower birth weight, and this is part of the low molecular weight phthalate. So it's consistent with the PCA models that we worked on. And most importantly was the fact that fathers and mothers and couples exposure to the total mixture reduced overall birth weight. But looking at the PIP, we found that it was possibly more the fathers that contributed to the overall effect compared to mothers, because the PIP was higher, and that's an indication that that the fathers contributed more to the hierarchical model that we applied. So in conclusion for the birth weight results, father's DHP and BPA mixture in the PCA model was associated with lower birth weight. Father's low molecular weight phthalates in the PCA model was also associated with lower birth weight. Independently of the other chemical mixtures, when they were held at the median concentration, MBP was associated with lower birth weight. And we found that father's total mixture concentrations across quantiles decreased birth weight in this study sample. For mothers, we found that BPA and the total mixture was associated with lower birth weight, and the couple's total mixture was also associated with lower birth weight. So what are the implications for this work? I guess that hopefully the message was, or the key message of this talk is that fathers and mothers both play a role in adverse reproductive outcomes. And in particular, with respect to birth weight and preterm birth, we have some evidence to suggest that fathers' exposure and mothers' exposure to DHP and BPA in particular might be detrimental to perinatal outcomes. Couples as a whole, the total mixture effect was associated with lower birth weight. And it seems that fathers might contribute more to birth weight than mothers with respect to this mixture analysis that we conducted. Most importantly, however, is that couples' preconception health is a modifiable exposure. And health promotion should begin before conception, and trying to reduce exposure among couples in the preconception window is a really important potential area of intervention that we are considering in terms of our next steps. And so we're trying to develop an intervention at the couple level to attempt to reduce exposure through modification of lifestyle and behavior, to reduce chemical exposure in the couple in order to try to impact reproductive success positively. And Kim's work, although it was in a different type of population, gives us hope that through altered lifestyle choices, we might be able to impact the concentrations measured in urine for mothers and fathers as well. And so that's one of our next steps that we hope to work on in 2021 and beyond. I think that concludes our presentation, but not without saying thank you to very important people on this team. So this work was a team-based approach. Yu Zhang took the leadership on this work, along with others on my team, including Vicente Mistelis, Yang Sun, Yixin Wang, Stelios Vagios, and Paige Williams at the Harvard Chan School of Public Health. I would also like to most importantly acknowledge the funding that I've received through an R01 for a different study using a similar cohort on perfluorinated chemicals that helps fund my work. And I'd like to thank the NIEHS program officers, as well as Antonia Califat from the Centers for Disease Control for her tremendous contribution to this work over decades. And then I can't forget the clinical people that have helped us recruit couples over the course of over a decade, John Petros and Irene Suter from the Massachusetts General Hospital Fertility Clinic. And if you have any questions following the seminar, we're happy to take them through email, and we could set up time to talk as well. So thank you very much. And I will turn the floor back to Hannah.
Great. Thank you both so much for that terrific presentation. And what we're doing now is having a little bit of a panel discussion. So if the three speakers could unmute and put yourselves up there, that would be great. I'm going to start with a question for Dr. Harley, and just wondering where you think the chemicals might be acting. Do you think they're acting at the ovary? Do you think they're acting at the brain? Where do you think that might be happening?
Yeah, that's a really good question that is going to get me in over my head really quick. But one of the things that we're concerned about with timing of puberty is starting the HPG axis. So that would be coming in from the brain. And when we're talking about, what we're concerned about, there's sort of two different kinds of puberty. There's our precocious puberty. You have your central precocious puberty that's just through the whole axis and would start up here, in the hypothalamus and the pituitary. And then we also see cases where girls have breast development, but it's not, that would be very localized, and it's not sort of systemic in that way. And what we're looking for here, and what we're suspecting and worried about, is effects that are on that HPG axis that are really kind of jump-starting that process. So you've got that HPG axis is just kind of, it's quiescent during all of childhood. It's active at birth, and then it goes to sleep, and then something makes it start when puberty starts, and we're thinking that it's happening through that, potentially through the brain. But again, the questions we're still trying to figure out.
No, it's really interesting. And I think the other thing that's an interesting part of the work that you're doing, but also the bigger story of earlier onset of puberty, is the whole association with increased body weight and increased body fat. So I have two questions around that. The first, I guess the first is there any association with some of the secular data in terms of the onset of puberty and historical concentrations of any of the things that you've been measuring? When was it that there started to be a big uptake in the use of plastics in particular? Does that kind of correlate with the changes over the last 40 years?
It's a really good question. We see, we've been exposed to endocrine disruptors over the last 40 years, but they're different, right? So 40 years ago, we were more exposed to things like DDT and PCBs. And as those really persistent ones kind of went down, what we're now seeing of the last 20 years has been an increase in things like BPA and phthalates and the phenols. And so I believe there has been an increase over the last 20 years or so. I think it's hard to pinpoint a secular trend. I think that the strongest secular trend that we see in parallel to the earlier puberty is the increase in obesity. I think that's really a major driver. But I want to point out that a lot of these endocrine disruptors we're talking about all seem to be linked to obesity. So there's sort of like, it's all kind of tied together. That phthalates were associated with increased obesity in childhood, and we also see that they're associated with increased earlier puberty in girls. So it's hard to tease all that apart.
Yeah. The one other question that relates to the effect of obesity, in fact, is how you didn't mention how you assessed breast development. So was that with, just a picture? Was that with?
No, because it was such a short talk, I kind of zipped over.
No, it's totally fine.
I apologize for that. We did tanner staging. I feel like we did an excellent job, if I can say so, gold standard tanner staging, where we did a clinical exam with palpation. So palpating the breast to determine the difference between adipose tissue and breast tissue, which is always tricky, especially in an overweight population.
And I think, the question isn't really settled, because tanner staging compared to breast ultrasound, which is really what's going to give you the idea of the different stages, is they don't agree very well, particularly in overweight girls. So we may not have the whole story. And
it's really challenging. Epidemiologic studies of onset of puberty are really
absolutely. Yeah. But it also does raise the question about onset of telarchy versus onset of menarche. And if we had really good breast assessment, would they match better? Just things like that would be really a helpful thing for us to understand. But this is really important work. I think the one other question that I might have for you just relates to the population. And you mentioned that this may be specific to the population that you have. And I'm wondering what other kinds of exposures might be unique to this population that may have either contributed or not contributed or messed up the analysis.
Yeah. This is a farm worker population, so they do have pesticide exposure. But we've measured many different chemicals on this population. And I'm trying to remember this. From what I remember, pesticide exposure and the personal care product exposure are not actually correlated. I believe we ran some sensitivity analyses looking at DDT, for example. But yeah, that's one of the things. I don't have evidence to say that there's some kind of systematic confounding because of this particular population, but this is a very distinct population, and we can't. Yeah.
But I mean, even having done the sensitivity analysis that you now had a chance to talk about, I think that's sort of a really helpful piece of your story. So again, so I'm going to save a couple of other kind of broader questions for all of you, but I'm thinking in terms of in terms of Dr. Masornian, in terms of the population of patients, just sort of knowing the MGH practice that this was done in, they do a huge amount of IVF. Were these IVF patients? Were they, yeah.
Great question. So yes, this particular study took place at the Fertility Center at the Massachusetts General Hospital. And just to describe the population a little bit more in terms of the clinical aspects, I would say about 60% of the births that we looked at were conceived through in vitro fertilization.
Okay.
30% were conceived through intrauterine insemination or other types of low technology.
Yeah. Okay.
And some of them were spontaneously conceived, but the majority of them were IVF conceived pregnancies.
And did that difference have any impact on any of your primary outcome measures?
So we do know from prior literature that IVF babies have a higher risk of preterm birth, low birth weight, and other types of adverse birth outcomes and pregnancy outcomes. In this particular group, we didn't find any differences between the general population in Boston, Massachusetts, or even the United States with respect to preterm birth. So these were singleton pregnancies, which I think we forgot to mention. They were all single embryo transfers. And the risk of preterm birth in this sample was 8%. And that's really actually on the low side in terms of what we see in Massachusetts, Boston, and other parts of the United States. So they were more at risk for these. And we did adjust for type of treatment in the model. So all the models were adjusted for treatment as well.
Were they adjusted for IVF versus not IVF?
Exactly. We adjusted for IVF. So ART versus non-ART. And ART we described both gamete manipulated in vitro. In addition to that, just to give some information, about 30% of the men had a male factor infertility, 30% had a female factor of infertility, and about 30% had what we called iatrogenic or unexplained infertility, which could be a number of different things. And this brings us some information. If we had a higher sample, larger sample, we could actually stratify by type of or cause of infertility. And the important part of this is that the preconception period, as I mentioned before, can really set up your general health. And so your health in your pregnancy is more than just your hormones, your ovaries. It's your general health, your inflammation levels, your immune function, your cortisol levels, all the other parts of your endocrine system are all tied into the other parts of your system, including your immune function. And so the preconception period is a period where, we have an influence because we have time, and we have time to be able to impact the outcome. And I think one of the messages that I'd like to share with clinicians and others, policymakers, is the importance of really targeting the preconception period from the couple point of view in order to improve the outcomes in birth, not even from birth, from even the embryo, from pregnancy loss outcomes, from miscarriage, from birth weight, preterm birth. And so focusing on the prenatal period, first of all, limits us to the woman, and focuses so much on the woman in the pregnancy and sort of adds blame to the woman, where the preconception period is about men and women, and trying to encourage sort of health promoting aspects where reduce weight, reduce smoking, reduce alcohol, reduce chemical exposure, reduce pesticide exposure through foods, increase fruits and vegetables, exercise. All of these things can improve our outcomes. And we need more studies to be able to see the impact of these sort of changes on outcomes. And that would be something.
So very quickly, so that we can get to the questions in our chat, I also wondered if you could address for us whether or not there was any kind of interaction with age, either paternal age or maternal age, with any of the mixture models. I'm just wondering whether that was something that you had an opportunity to look at, Dr. Zhang.
Yeah, Yu Zhang, that's your question.
For the age, yes, but we didn't adjust. We adjust for age as a continuous variables in our models, but we did not add it in the model to test its interactions in the mixture.
The reason I'm thinking about that is that you sort of have an opportunity to separate the paternal from the maternal influences just because of the developmental aspect of the male and female gonad, right? So the female gonad is likely to have, accumulated trauma, accumulated things that affect ovarian aging over time. Whereas for spermatogenesis, there's this constant renewal process that really means that it's much more of something that you can make change in, which is also why I think the
Yeah.
results of your study are so compelling, because I think, sort of, anyway, it would just be really interesting to look at that.
Yes, that's very interesting. Maybe we can stratify the results by the age, because adding age as another term in the model may be too complex for the mixtures.
Yeah,
But stratifying it may be interesting.
I would have to leave that to you.
I would like to add just a small comment to that, because I think it's a really critical point that Dr. Hall brings up. And I actually had a discussion with this with one of my students in my course last semester about the fact that, so just briefly, the ovaries, the gonads, the female gonads age over time, like Dr. Hall just said. And what we've noticed in some of our work is that the younger females, the women that are less than 35 years of age, the impact of the chemicals contributes or impacts the outcome of those of the ovulatory cycle more so than the older women. And what we speculated here was that by the time you age, there's already a hit on the ovary and how it functions, and the impact of the chemicals may just do a little bit more damage to the ability for that ovary to reproduce or to ovulate. But among younger women, there's more of a buffer, there's more of a threshold in which these chemicals can actually cause reduced ovulatory potential. And we wondered whether or not, I think you raised a good question, that perhaps similarly with these birth outcomes, that maybe age already had an effect on these potential adverse outcomes. And so the younger women may be actually more at risk.
Right, and I think you don't know until you take a look at it, because there may be an assumption that, again, as we've heard from both of the talks, that what you're measuring is a moment in time, but that moment in time may be reflective of a lifelong, habit of certain kinds of behaviors. So I think what I'll do, because I was going to ask about the most important things that you thought about in terms of, how we could go about making these changes, but I think that you all addressed this really nicely. And so just in the interest of time, I'm going to turn the mic over to Karen, and Karen's going to go through the Q&A, and hopefully we'll be able to. Karen, are you fine in terms of knowing who to address it to?
Yes. Great.
Okay, great. Perfect.
Thank you so much, guys, for joining us today. I've gone through the questions and sort of grouped some of them together. Dr. Harley, we had a couple questions about exposures. What might account for the mother's variance in different toxicant exposures? And then were you able to see if, these exposures correlated across time? People who are exposed in utero, were they also in high exposure when you looked at age 9?
So the first question is, what might account for their exposures? So specifically, we're talking about the low molecular weight phthalates, the parabens, triclosan, and BP3. And we know that the main source of exposure to these chemicals is personal care products. It's not necessarily the only source of exposure, but it really is the main source. So we're really thinking that that's probably, the differences in exposure are probably coming from their different product use. There might be cleaning products in the home, or, there are some other possible sources there. I believe you can get parabens through food, but the primary exposure is personal care products. Now, when we started our study 20 years ago and enrolled these pregnant women, we were really looking at other chemicals. And so we didn't gather information about all the personal care products they were using. If we'd known that 20 years later, we would want that information, but we didn't have it. So we can't specifically look at their personal care product use to correlate it to, but we went back and we did a different study on teenagers. And in that case, it was really clear that their variability and their exposure was due to personal care products. You also asked how well these track over time. I can't really answer that with our data, because we really only looked in the moms during pregnancy and then the kids were years old. And there's not much correlation between what the moms were having, in pregnancy, and then 9 years later what the kids had. So unfortunately, I just can't really speak to that.
Okay, great. And we also had, a question about just replacement in alternate chemicals. We're seeing some move away, for example, in the triclosans, but, what have you been able to look at that over time? Just what chemicals are replacing the ones that maybe you were originally studying. And then we had another question specifically asked about quats, as they are in many cases replacing triclosans as antimicrobials, and they are being used a lot right now during the pandemic.
Yeah. Okay, those are two questions. Let me see if, what was the first one again?
That's just the shifting, just the
yes. Okay, so that's, I mean, that is such a good point and such a difficult thing for us. So think about our study. We followed kids from in utero through puberty. So that's like a 15-year study. And so when we enrolled the kids, you know, we took all those prenatal samples were taken in the year 2000. And these replacement chemicals weren't being used in 2000. And so it's one of the frustrations, I think, of being a reproductive epidemiologist in this field is that by the time we get the research done, the industry has moved on. And so now it's true. We're definitely seeing, you know, triclosan has come out, MEP levels are going down, other phthalates are coming up, and it takes us so long to do the epidemiology that we're kind of always playing catch up. But it's really, really critical. And that's definitely what we want to do now is look at what the replacement chemicals are. Are the replacement chemicals safer? Are there some of the same concerns? Yeah, that's definitely something that we need to be thinking about. The question about quats, yeah, so quats are used in personal care products and cleaning products both, I believe. And I haven't seen very much epidemiologic research on them yet. But they're, you know, this is what we're saying. These are the things that we need to be starting to focus on now, definitely.
Definitely. Yeah, we had a couple of questions since you talked so much about the timing of puberty. Can you just talk a little bit about, you know, what do we know about how the timing of puberty affects health later in life?
Yeah. So one of the reasons that this is important is that girls who enter puberty earlier are at increased risk of breast cancer and other reproductive cancers later in life. So there's this whole life course of earlier puberty affecting reproductive cancer much later in life. It also has a lot of psychological and kind of cultural effects on girls. If you enter puberty earlier, it puts you at increased risk of risk-taking behaviors in adolescence and also mental health problems in adolescence. So there's kind of immediate psychological issues and then this kind of long-term reproductive concerns about early puberty.
Okay,
related to breast cancer.
Dr. Masarlian, we had a couple questions about you know, interventions. You know, one was about the timing of interventions before conception. Do we know anything about, you know, how much time you would want before conception? And then relatedly, we had a couple of questions about, you know, many pregnancies are unplanned and what advice should we provide for healthcare providers and for women?
Those are wonderful questions. Thank you so much to the audience for bringing those to our attention. So in terms of the window of time before conception, we don't have a great answer for that because there's not been intervention studies in this area, but we could use reproductive biology to sort of inform this a little bit. And we know, at least from the father's exposure point of view, that spermatogenesis takes 90 to 120 days to happen. And so that might be a particularly important window of time for fathers to reduce exposure to chemicals, increase healthy lifestyle choices. And using, and I and people on my team looked at sort of similarly looking at paternal windows of exposure specifically around spermatogenesis versus before spermatogenesis and whether or not there was differences in father's outcomes for preterm birth and birth weight. And we did find some indication that that window of time might be the one that's important for these outcomes, the window of spermatogenesis. As far as for women, well, we know that we're set with the ovaries that we have, with the eggs, with the oocytes that we have at birth, and there's attrition over time. There's aging of those ovaries and the oocyte primordial follicle count over time, and the recruitment of those primordial follicles to secondary follicles is impacted by age, and the number of follicles that we recruit per month per cycle is impacted by age. So we can't do much about what we're sort of set with when we time our pregnancies, if we do time our pregnancies. But we know that the cycle of conception might be particularly important. And we've shown this from some of our previous work, that it was exposure around the time of conception, around the periconception window, the time of ovulation, the time of fertilization, implantation. That might be really an important critical window for these outcomes. And the reason why this might be is that there's different markers on the oocyte that signals to the sperm, that matches up with the sperm, that then creates the embryo. And all those little epigenetic changes that happen, there's a demethylation or remethylation of the embryo, of the early stages of the embryo, that could be impacted at that exact time of periconception. So the actual cycle of conception might be very important for women. And inflammation in that period of time, inflammatory biomarkers, cytokines, immune function might impact the implantation success, and how those processes take place throughout pregnancy may set up the pregnancy to be adverse from very, very early on. And so that might be an intervention point. But to comment on the fact that this is really pregnancy planners, indeed, that's correct. Infertile or subfertile couples are attempting pregnancy. They have time and energy to invest towards reproduction, and most pregnancies, a large percentage of pregnancies, are unplanned. One particular area for clinicians and people seeing women, treating women, I think that we're really interested in pursuing is when women come in with miscarriages or pregnancy losses. Some of these are pregnancies that were not planned. And so that's an opportunity to talk to women about preconception health. If they, if they have a pregnancy loss on an unplanned pregnancy, but maybe planning a pregnancy in the future, that could be an opportunity to intervene and talk to women about preconception health. And then that's a way of accessing that population of unplanners. And usually pregnancies that were unplanned are a different group of women altogether in order to tailor interventions to. And that might be one way of accessing that population that we've considered in some of our work. Translate to evidence for clinicians.
Definitely. We had several questions that I guess either of you could answer about timing of puberty and oocyte quality. Do we know anything about that?
Oocyte quality. So Kim, do you want to answer this?
No, it's something that I don't feel really qualified to answer that, but if you have comments, that would be.
I don't have any concrete answers to it. The only answer that I would have is that the primordial follicle recruitment to the secondary follicles is a process that happens cyclically, monthly, during menses, during ovulation for women. And there are different things that can impact that recruitment of those follicles. Maybe Dr. Hall could answer this better than us in terms of how the primordial follicles get recruited to secondary follicles at each cycle, and the number of follicles that get recruited, and then how they proliferate and mature, and then one ovulates and becomes follows into the fallopian tube. So pubertal timing, I don't think there's any evidence per se, at least in humans, that shows that pubertal timing might impact that. But the more cycles you recruit from, so the earlier pubertal onset, your attrition of those cycles is more. So you have more cycles across your reproductive lifespan, and that impacts things like cancer, but it could also impact possibly fertility, or possibly, what we would consider reproductive aging, because there's only possibly a finite number of cycles in which you can ovulate in your lifetime. That's something that's not been studied fully yet, but we are looking at DNA methylation as a potential marker of fertility age that we're working at in my lab as well. So looking at a fertility clock using similar things like the epigenetic aging clock to be able to assess reproductive age in women through biomarkers, DNA methylation biomarkers. But there's not really good data yet on that. But
yeah, I was gonna just jump in and say that again, there just aren't good data that address that specific question. But one of the things to take away as well, Carmen, is just remembering that so much of the loss of ovarian follicles and the recruitment into that doesn't have to do with menstrual cycles at all. It has to do just with the number of years an individual has been on the planet. So there's this sort of inexorable loss of follicles and oocytes that throughout our life. Certainly cycles contribute, but the amount they contribute is pretty minuscule. A little bit more after the age of 35. And so where you get actually each cycle, in fact, there's an increased loss from that pool. And so both of those things need to be taken into account. But I think it's a really interesting question. And I do not know the answer to it.
Great. We're definitely over time, but there are a couple of questions about race and social economic status that I wanted to address. We had one audience member point out that presumably at the IVF clinic we're talking about couples from higher socioeconomic status, and then Dr. Harley, your population were farm workers, and then we had another audience member talk about how Black women are also exposed to many EDCs in their personal care products that are marketed towards them. So could you guys comment about, racism and socioeconomic status and how you think it interplays with some of the things that you're looking at?
Yeah, I'd love to talk about that. We're actually working on a study right now that's looking at personal care products that are marketed to Latino women, Asian women, and Black women as those separate groups, to look at the ingredients in those products and find out, how differential the exposure and what kind of different chemicals are being exposed, women are being exposed to. One of the things that I've noticed working in this field for the last 10 or more years, specifically looking at personal care products, is that when I started doing this more than 10 years ago, hardly anybody, it wasn't on anybody's awareness, people's consciousness that there were EDCs in personal care products. And then we really started to see kind of an awareness, particularly in white women. It became a very kind of upper class kind of issue. There were a lot of low chemical products, but they were very expensive. So this kind of economic gradient. But what I've noticed in the last 5 years or so is just a much more sort of broad reaching awareness of chemicals, and particularly among African-American women. I think there's been a real awareness about hair products, for example, that are really marketed towards African-American women and what's in them. And just looking from a marketing point of view, I'm seeing more and more products that are really, particularly in the African-American space, but other markets, that are really starting to advertise that they don't have these chemicals in them. So I'm seeing a consumer shift. It's interesting because there hasn't been very much regulatory shift, but there has been very much a shift, consumer-driven shift in what products are available to many different populations. We're getting there.
And Carmen, I mean, you talked about the importance of, couple studies. Are you aware of, ways that we could get more diversity in some of these studies that you describe?
I think that concern is an important, relevant one. The IVF population, at least at the Massachusetts General Hospital, is primarily white. And that's, fairly consistent with the general population in Boston. So it's kind of a representative sample. When we compare sort of socio-demographic indicators in the sample and in the clinic and then in the population of Boston, we're not oversampling or undersampling any specific group. It's sort of consistent. But it is a study of mostly white people, white couples, and that's a big disadvantage in terms of us being able to generalize the findings to others. These findings have limited generalizability to other groups of people that are not represented through that cohort. It's challenging for us given the setting, but we do have plans to try to over-enrich some of our enrollment in the next phase of the study. We're working on trying to provide more direct recruitment and attention to couples that come in from different racial and ethnic backgrounds. But apart from being able to do that, it's challenging for us to change the demographics in which the clinic resides. That said, I think, helping other researchers in different parts of the United States who have different demographics to start a cohort, or at least work on a similar cohort, maybe borrow some of the materials and work and translate some of the materials and work that we've done, where they could leverage the investments that we've made for them to collect data that might be more diverse. Might be a way of us pooling the data, which would be interesting. That's that I've had conversations with some of my other colleagues in different parts of the United States.
Yeah, great. Thank you so much all of you for coming to speak with us today and share your research. Hannah, I'll turn it back over to you.
Great. Thank you so much, Karen, and to all of you. We're approaching the end of today's webinar. A video recording will be available on Che's website soon, and tomorrow you'll receive an email containing a link to the video. The next Generation Chemical Partnership webinar is on prenatal exposures and fetal outcomes. It will take place on February 25th at 1 p.m. Eastern Time. To learn more and to RSVP, please visit our website at healthandenvironment.org. If you are new to Che and would like to stay updated about upcoming events or more, please sign up to receive our newsletter by selecting the Join Us tab at the top of any page on our website at healthandenvironment.org. Additionally, if you appreciate these Che Partnership webinars bringing you the latest environmental health research for free, we encourage you to support Che's ongoing work by making a tax-deductible donation via our secure website. Again, our website is healthandenvironment.org. With that, I would like to thank our speakers and moderator, Janet, Kim, Carmen, and you for taking time to present today, and to you, Karen, for your excellent moderation during the Q&A. Thank you so much for joining us. We hope everybody stays well and healthy. Take care.